So now we have seen fatty acids, classification of fatty acids, and how they are absorbed and how they are transported to different tissues. Okay. Now we are going to see how they are oxidized. So once they transported to cells, so this fatty acid may be utilized to synthesize plasma membranes to synthesize a kind of fat molecules as a deposit molecules, and cells can oxidize this fatty molecule to get energy from it. Okay. So and then we are going to see about the ketone bodies also. Once fatty acid degrade, um, oxidized, what are the end products and how they are converted into type of ketone bodies, and what are the uses and what are the disadvantages with these ketone bodies we are going to see now. Okay, so now the breakdown of fatty acid to acetyl-CoA units. So we have seen breakdown of glucose to pyruvate. Okay, here the fatty acid breakdowns to acetyl-CoA molecule, acetyl molecule. Okay, so that we can call as glycolysis of fatty acids. This we can call as glycolysis of fatty acids, and strictly aerobic conditions. So oxygen is required in this, and this reaction occur in mitochondria. Okay, glycolysis of glucose occur in cytosol, whereas glycolysis of fatty acid degradation occur in mitochondria. Okay, the the molecules which is formed here, acetyl-CoA molecule, is directly a kind of substrate molecule in TCA cycle, that is Krebs cycle. We have seen that one. Okay, so overproduction, over oxidation of fat molecule, over oxidation of fatty acid leads to generation of acetyl-CoA molecule, and then it converts into ketone bodies. Okay, that's why we can call as overproduction causes excess of ketosis, ketone bodies generation. Okay, so exemplified of aerobic metabolism, and it is most powerful phase. So most powerful phase means most energy yielding phase. That's why it is called as most powerful phase. You can see lots of ATP molecules generate from the oxidation of fatty acid molecule inside the cell. Okay, so now how it starts fatty acid oxidation. Fatty acid oxidation must be esterified with coenzyme A. So that is activation of fatty acid. We can call as activation of fatty acid leads activation by esterifying the coenzyme A. Okay, before they undergoes for oxidation of the oxidative degradation, are utilized for synthesis of complex lipids, or attached to other proteins like become lipoproteins. So before all these steps, it has to activate it. Fatty acid molecule has to activate it, and that activation will be done by esterification of the coenzyme A molecule. Okay, the enzyme, the esterification part will be done by enzyme which is called as Acetyl-CoA synthase. Okay, thiokinases, otherwise we can call as Acetyl-CoA synthesis, are the enzymes. Those use coenzyme A molecule and attach to fatty molecule, free fatty acids. Those can be used for oxidation, or those can be used to synthesize complex lipids, or those can be attached to a protein to become lipoproteins. Okay, so these acetyl-CoA or thiokinases are present on endoplasmic reticulum or outer mitochondrial membrane and catalyze the activation of fatty acid to become acetyl-CoA molecule. Okay, so this process is ATP rich or ATP dependent pathway. So occurs in two steps. Okay, so the important point to keep in mind, there are different types of acetyl-CoA synthesis for fatty acids of different chain of length. Okay, so only one, you don't think only one acetyl-CoA synthase is enough to synthesize or esterify all type of fatty acids. Okay, so there are different types of these enzymes. That difference depend on the chain length of fatty acid. Okay, we have seen short chain, medium chain, and long chain fatty acids like that. So thiokinases or acetyl-CoA synthetases also differ based on the chain length. Okay. So here this occur in two steps. You can see Fatty acid interacting with the ATP molecule, which cleaves pyrophosphate. Subcutaneous, subsequently, this pyrophosphate will be converted into inorganic phosphate later with pyrophosphorylase, and then this acyl adenylate, which is having a fatty acid molecule, interact with the coenzyme A in the presence of thiokinases that leave the AMP and coenzyme A attached to the acetylide to the fatty acid molecule and become Acetyl-Coa here. Okay, so it is two-step process. First, fatty acid interact with the ATP molecule and convert into acyl adenylate, which is cleaving inorganic phosphate, as we seen in previous slide. And acyl adenylate interact coenzyme A in the presence of thiokinases, and which esterify the fat molecule to Acetyl-Coa and leaving the ATP molecule. So here, overall reaction, fatty acid molecule plus ATP plus coenzyme A, you will get Acetyl-Coa plus AMP plus 2 inorganic phosphate. Okay, 2 inorganic phosphate, initially you will get pyrophosphate, and that will cleave into 2 inorganic phosphates. That's why you get 2 inorganic phosphate molecule. Okay, so now fatty acid is activated in the presence of ATP. So to activation, that ATP cleavage is must here. So that's why it is get activated by the acylation. Okay, so now, so to get into our degradation, oxidative degradation, the fatty acid, all enzymes related to fatty acid degradation present in the mitochondria matrix, which is, which mitochondria is double membrane layer, and the outer layer you can see, and the inner layer you can see the jelly-like structure inside we call as matrix. Okay, so fatty acid Acetyl-Coa formed in cytosol by enzymes of outer mitochondrial membrane and endoplasmic reticulum. So here the enzymes Acetyl-Coa thiokinases are present on the outer membrane of the mitochondria and endoplasmic reticulum. So that means Acetyl-Coa are present in the cytosol. Okay, so it has to transport. So it has to transport inside the mitochondrial membrane, inner membrane, and it has to pass from outer membrane and then inner membrane and then enter into matrix. Okay, so free fatty acid breakdown in mitochondrial matrix via beta oxidation, that is acyl activated free fatty acids. For most steps of pathway, there are multiple enzymes specific for particular fatty acid chain length. Okay, as I said in previous class, slide, activation acetylkinases, Acetyl-Coa synthesis, and thiokinases. Okay, those are present, in different forms here. Different isoforms will be there, depending on the side chain, okay, length of the chain. Okay, so this oxidation also, different type of oxidation, different type of enzymes will be present. Those based on the fatty acid chain length. Okay, many enzymes are soluble proteins located in the mitochondrial matrix. Those are involved in the beta oxidation. But enzymes specific for very long chain fatty acids are associated with the inner membrane facing to matrix. So if you see the long chain fatty acids, those enzymes will be attached to the inner mitochondrial membrane and they will be facing towards the matrix. So they will be attached, they will be anchored to a mitochondrial inner membrane, and then they will remaining rest of the part will be facing associated with the matrix. So fatty acid Acetyl-Coa formed outside can pass through outer membrane, mitochondrial membrane, but cannot penetrate inner mitochondrial membrane. So the Acetyl-Coa, which is formed outside the mitochondrial membrane, can easily pass this outer mitochondrial membrane, but it cannot pass the inner mitochondrial membrane. To enter into that, it requires one kind of transporting protein, which is called as carnitine. Okay, transfer of fatty acid moiety across the mitochondrial inner membrane requires carnitine protein. Okay, so carnitine palmitoyl transferase. So carnitine palmitoyl means here This is carbon 16 fatty acid. If it is 18 fatty acid, aloic acid, carnitine enolide transferases, or if it is arachidonic acid, carnitine arachidonic transferases, like that. Okay, so that kind of different types of enzymes will be there. So here, carnitine is the protein that will attach to Acetyl-CoA in the presence of carnitine palmitoyl transferases. Palmitoyl means fatty acid transferases, so that will remove the ester coenzyme esterification. So now you will get the fatty acylcarnitine. Okay, fatty acylcarnitine, the inner mitochondrial membrane will have a carrier transporter. Okay, an antiporter, which is called as antiporter. So here you can see in the cytosol, Acetyl-CoA is formed and carnitine palmitase is present, cytosol as well as present here. So this interact with Acetyl-CoA and makes carnitine acyl molecule. So this carnitine acyl molecule interact with the antiporter and it will be transferred inside the through the inner mitochondrial membrane inside the matrix. Okay, so now here acylthiokinesis present in mitochondria matrix, which uses this carnitine acyl molecule and esterify the acyl coenzyme A to that acyl carnitine acyl molecule, carnitine fatty molecule. To now you can get Acetyl-CoA molecule here. Okay, so the free carnitine will again transported outside and which will recycle the process here, cycle the process here. Okay, so carnitine palmitoyl transferase to enzyme, and enzyme within the matrix transfer the fatty acid from carnitine to Acetyl-CoA here. Okay, sorry. I said thiol kinase, that is not thiol kinase. This is carnitine palmitoyl transferase to enzyme. This enzyme transfer the carnitine and exchange the Acetyl-CoA molecule with carnitine. So free carnitine will form here. This free carnitine again back to outside, and then carnitine palmitoyl transferase 1, which use this free fatty acid Acetyl-CoA molecule and then ligate it with the carnitine molecule. So like this, it will cycle reaction. So Acetyl-CoA molecule enter into mitochondrial matrix, where it will beta oxidation will take place. Okay, so control of fatty acid oxidation is accepted mainly at the step of fatty acid entry into mitochondria. So you can see fatty acid oxidation normally, if you stop this process, if you stop this transportation from outside to inside, then you can easily control the beta oxidation of fatty acid, and also you can usually reduce the ketone bodies generation here. Okay, so for that, methylmalonyl-CoA, which is also precursors of fatty acid synthesis, inhibit the carnitine palmitoyl transferases, which is present outside to make carnitine fatty acid. So that inhibit this one, so that it will not transport it inside the membrane. Okay, methylmalonyl-CoA is produced from the acetyl-CoA by the enzyme which is known as acetyl-CoA carboxylase. So this acetyl-CoA molecule normally after oxidation, beta oxidation, acetyl-CoA molecule will present. So in the presence of acetyl-CoA carboxylase, carbon molecule will be ligated to this molecule, so it becomes methylmalonyl-CoA. So methylmalonyl-CoA is the competitive inhibitor or regulatory inhibitor for this carnitine palmitoyl transferases 1, which is present in the mitochondria outer membrane or cytosolic side. Okay, any doubts so far? Any doubts so far?
No, sir.
Okay. So now acetyl-CoA molecule is present inside the mitochondria matrix, and it can degrade into acetyl-CoA molecule. It can start the cleavage of fatty acid, degradation of fatty acid in the form of acetyl-CoA molecule. So what we require here? Okay, so here you can see fatty acid structure. So what we have told that carboxyl carbon and carboxyl carbon, next carbon we call as alpha carbon, and next carbon we call as beta carbon, and last methyl end we can call as omega carbon. That's what we have seen in the classification. So now this we call as beta oxidation. So the oxidation cleavage occur at the beta carbon molecule. That's why it is called as beta oxidation. So here you can mark here one which is acylated with the Acetyl-Coa, and next carbon to carboxyl group which is called as alpha. That third carbon which is called as beta carbon. So here beta carbon, between beta carbon and the alpha carbon, the carbon cleavage will occur. That's why it is called as beta oxidation. So here you can see different types of enzymes, dehydrogenase enzymes. Okay, as we seen in previous classes, those enzymes which remove the hydrogen or hydrogen or electron molecules, those are called as dehydrogenases. So here, one enzyme, Acetyl-Coa dehydrogenases, characterize the oxidation of fatty acid moiety of Acetyl-Coa to produce double bond between carbons atom 2 and 3. So here you can see alpha carbon and beta carbon atom. So it will remove the hydrogen ion molecules, and because of that you can see double bond formation between alpha and carbon, alpha and beta carbon. Okay, which is known as trans delta 2 enoyl CoA. Okay, so there are different types of Acetyl-Coa dehydrogenases in the matrix. That's what I told you. Different types of enzymes will be present which are involved in the oxidation process. Those depend on the chain length. Okay, there are different types of Acetyl-Coa dehydrogenases for short carbon fatty acid like 4 to 6, medium size 6 to 10, and long, very long like 8 to 12 and 20, 24 like the fatty acid. Okay, so very long fatty acid dehydrogenases is bound to the mitochondrial membrane. The others are soluble enzymes located in the matrix. So very long chain fatty acid, if you go for beyond 2022, so those enzymes, dehydrogenases enzymes are ligated, are linked with the mitochondrial, inner mitochondrial membrane, and they will face towards the matrix. Other enzymes are soluble in matrix. Okay, so matrix is jelly-like structure, as we see, know that is looks like cytosol inside the cell. So here the hydrogen molecules, the hydride ion molecules are removed, and these hydride ion molecules are accepted by FAD molecule. So FAD molecule is an acceptor, and in the presence of Acetyl-Coa dehydrogenases, FAD become to FADH2 molecule. Okay, so the mechanism, glutamate side chain carboxyl extract proton from the alpha carbon of the substrate, facilitating transfer of 2 electrons with hydride ion from the beta position to FAD molecules. So the reduced FAD accept to second hydride ion yielding FADH2 molecule. So the reactive glutamate and FAD are on the opposite sides of the substrate at the active sites. Thus, reaction is stereospecific, yielding trans double bond in enoyl CoA. So here, this glutamate and FAD is opposite side at the active site. So they will remove the hydrogen molecules specifically. Okay, specifically means, we have seen trans and cis configuration. So specifically, when they remove, you will get a kind of trans orientation, trans type orientation. So here, trans, that's why it is called as trans delta 2 enoyl coenzyme A. So now next, the trans delta 2 enoyl CoA in the presence of enoyl CoA hybridase. Hybridases are the enzymes, those involved in the hydration of the substrate molecule. So they will provide the water molecule. So it makes double bond hydrated. It makes double bond into single bond by adding water molecule here. So now you will get 3L hydroxy acyl CoA molecule. Okay. And third step, another dehydrogenase enzyme activity involved. Here, the hydride ion acceptor is NAD plus molecule. So, this reaction, will remove the hydrogen ion molecule from the beta carbon atom. So you will get ketone molecule here. After removing hydride ions, NAD is accepted. So NADH plus H molecule will generate here. So ketone molecule, which is called as ketoacyl, beta ketoacyl group molecule is formed here by the action of hydroxyl-CoA dehydrogenase enzyme. Okay, so next beta ketothiolases, which is keto, which esterified the thiol group, acyl-CoA molecule here. So it breaks the carbon-carbon bond between alpha and beta carbon molecule, and the coenzyme A will be acylated here. So you can get acetyl-CoA molecule carboxyl group and the alpha carbon cleavage, and coenzyme A will be attached already there. So another coenzyme A will be attached to the beta carbon. So you will get a fatty acid, 2 carbon shorter fatty acid cell group, and another one is acetyl-CoA molecule. Okay, so this is the way beta oxidation occurs here. Okay, at the fourth step, which keto acid, which is higher beta keto acyl-CoA. The fourth step, beta ketothiolase, what we see in the breakdown of carbon-carbon bond, breakage of carbon-carbon bond. The mechanism here is cysteine A attack on the beta keto carbon. Acetyl-CoA is released, leaving the fatty acid moiety in thiol ester linkage to cysteine thiol. Okay, thiol SH coenzyme A displays the cysteine thiol, yielding fatty acid acyl-CoA molecule. That is 2 carbon less fatty acid. We will get 2 carbon shorter fatty acid plus acetyl-CoA molecule because of carbon-carbon bond cleavage between alpha and beta carbon. So this is a membrane-bound trifunctional protein complex, which 2 subunits types expresses the enzyme's activity for 2 to 4 of beta oxidation pathway for long chain fatty acids. Equivalent enzymes for shorter chain fatty acids are soluble of mitochondrial matrix. So summary of this beta oxidation, you can see one round of beta oxidation. You can see fatty acid molecule, which enter into mitochondria matrix. It may be a carbon 16, carbon 14, carbon 18, like that. So this fatty acid, acyl-CoA plus FAD plus NADH plus coenzyme A, you will get one round fatty acid acyl-CoA molecule, which is 2 carbon less fatty acid plus FADH2 plus NADH plus H plus ions plus acetyl-CoA molecules. Okay, so this beta oxidation is cyclic. So one round is over. Next, this molecule become substrate for the further round. So the product 2 carbon shorter is the input for the another round of this pathway. Okay, so if as is usually case, the fatty acid contains an even number carbon atoms, the final reaction cycle butyl-CoA is converted into 2 copies of acetyl-CoA molecule. So 4 carbon. Finally, you will get 4 carbon molecule, which is called as butyl-CoA. So if it is even number, so now from that you will get 2 copies of acetyl-CoA at the last. Okay. So acetyl-CoA molecules are the end products after beta oxidation. All acetyl-CoA molecules are entered into Krebs cycles to yield additional NADH plus molecule and FADH2 molecule and ATP molecule. Okay, so you can see more NADPH molecules are formed by cleaving the carbon-carbon bond in the fatty acid. FADH2, NADH2. Again, this acetyl-CoA molecule, if enter into Krebs cycle, it will give rise more NADH plus molecule and FADH2 molecule and ATP molecules. So fatty acid oxidation is the major source of the cell ATP. So different types of diseases identified when enzymes get mutated in this fatty acid oxidation. So the plasma membrane fatty acid transporter, which is called as CD36, okay, which is present in inner mitochondrial membrane. If it is get problem, that will give some kind of problem in the beta oxidation. And carnitine palmityl transferase 1 and 2, which is transfer Acetyl-Coa molecule into carnitine acyl molecule outside the membrane and inside the membrane. And Acetyl-Coa dehydrogenases for various chains of length fatty acids. Hydroxy-Acetyl-Coa dehydrogenases for medium and short chain fatty acid. Medium chain beta ketothiolases, which is fourth step enzyme. The trifunctional protein complex, which is larger protein molecules. Electron transporter flavoprotein, which is involved in the electron transport chain. Okay. So human genetic diseases, symptoms are depending on the specific genetic defect, but may include hypoglycemia and failure to increase the ketone bodies production during the fasting. Fatty acid degeneration of liver, heart and skeletal muscle defects, maternal complications of pregnancy, sudden infant death also you can see because of this enzymes mutation. Okay. Hereditary deficiency of medium chain acyl-CoA dehydrogenase, which is called as MCAD, the most common genetic disease related to fatty acid metabolism, has been linked with the infant sudden death. Okay, during the pregnancy period. Okay. So that is about fatty acid oxidation. So we have naturally all fatty acids are even number fatty acids. Okay. And saturation fatty acid and unsaturation fatty acids. Okay, so if you have odd number fatty acid, okay, if you get the double unsaturated fatty acids, okay, then in that case, additional enzymes deals the catabolism of fatty acid with an odd number carbon atom or with the double bonds. Okay, additional enzymes will work on this type of odd number fatty acids and unsaturated fatty acids where double bond is present. The final round of beta oxidation of fatty acids with an odd number of carbon atoms yields acetyl-CoA and propanyl-CoA. Okay, so acetyl-CoA is 2 carbon molecule, propanyl-CoA is 3 carbon molecule. Okay, propanyl-CoA is converted into Krebs cycle intermediate into succinyl-CoA by the pathway involving the B12 vitamin. Okay, double bond. Okay, mostly naturally occurring fatty acids have cis configuration. Okay, if you have trans fat, okay, so you will trans fats also enter into double bond beta oxidation. C carbon atoms are removed 2 at a time. The double bond may end up a wrong position or wrong configuration to be corrected. Substitute for acyl-CoA hybridase. Okay, that is about beta oxidation occurring in mitochondria. Any doubts so far?
No, sir.
Okay, so beta oxidation major site is mitochondria, but however, some part of mitochondria, part of beta oxidation also occur in peroxisomes. So peroxisomes are single membrane organelles inside the cells. Okay, so they have enzymes, different purpose. Okay, like including catalysis and different enzymes which involved in the degradation of hydrogen peroxides. Okay. So beta oxidation for long chain fatty acids. Okay, long chain fatty acids occur in peroxisomes. FAD is the electron acceptor for peroxisomal acyl-CoA oxidases, which catalyze the first auxiliary steps of the beta oxidation pathway we have seen in the mitochondria. Okay, So serious genetic diseases are associated with defects in deficiency of enzymes of the peroxisomal beta oxidation system. Okay, if mutations occur in peroxisomal beta oxidation enzymes, serious genetic diseases will occur. Peroxisomes also contains enzymes for essential beta oxidation pathway that degrade the fatty acids having methyl branches, such as phytonic acid and breakdown product of chlorophyll. Wherever branch chain fatty acids are there, those specific enzymes also present in the peroxisomes. So you can see advantages with the peroxisome oxidation. So long chain fatty acid, cholesterol. Cholesterol is also comes under very long chain carbon fat related molecule, compound fatty acid. So now beta ketone bodies. So once the fatty acid degrades acetyl-CoA, if more fatty acid degradation occur inside the cell, more acetyl-CoA molecules will form. More acetyl-CoA molecules really present inside the cell. They may enter into Krebs cycle or may convert into a kind of molecule which is known as ketone bodies. Those are acetoacetate, acetone, and beta hydroxybutyrate. Okay, so this acetyl-CoA molecule may enter into fatty Krebs cycle. If not, so they will convert into acetoacetate molecule or acetone and beta oxidation, beta hydroxybutyrate molecule. So these three molecules we call as ketone bodies. Okay, so these ketone bodies, when they are present in more, they are soluble in blood and can be transported into various types of tissues, and again they regenerated into acetyl-CoA molecule, and acetyl-CoA molecule gives the energy via PCA cycle. So almost all organs will use this acetone or ketone bodies for the energy generation purpose via Krebs cycle. After they convert back to acetyl-CoA molecule inside the cell. Okay, so here you can see in glucose oxidation to pyruvate to acetyl-CoA. So if you see acetyl-CoA can form from fatty acid, and if more acetyl-CoA molecules are there, it will convert to ketone bodies, and the acetyl-CoA molecule is also substrate for the cholesterol synthesis. So if acetyl-CoA molecule more, it can convert into ketone bodies or it may enter into Krebs cycle. That's what we have seen. Okay, so during the fasting of carbohydrate starvation, oxaloacetate is depleted in the liver due to gluconeogenesis. This impedes the entry of acetyl-CoA molecule in Krebs cycle. Acetyl-CoA in liver mitochondria is converted into them to ketone bodies. Okay, so when we enter into fasting condition, when we are in fasting condition, oxaloacetate will goes down. Okay, so once oxaloacetate goes down, that block the acetyl-CoA molecule and enter into Krebs cycle. Okay, so then ketone bodies, which is generated from fatty acid degradation, and then it will convert into second route by second route into ketone bodies. Okay, so acetyl-CoA in mitochondria is converted into them ketone bodies like acetoacetate and beta hydroxybutyrate. So now you can see how acetyl-CoA molecules are generated here. So one acetyl-CoA molecule joins with a second acetyl-CoA molecule. So in the presence of trial kinase, okay, trial kinase, it forms as an acetoacetate molecule. This is one type of ketone body, and this acetoacetate, when it joins with the third acetoacetate molecule in the presence of HMG-CoA synthase, so you can get HMG-CoA. HMG-CoA is nothing but beta hydroxygluteryl-CoA. So this HMG-CoA in the presence of HMG-CoA lyase, it will give acetoacetate and acetyl molecule. Okay, so this acetoacetate later stages can convert into butyryl hydroxybutyrate in the presence of beta hydroxy dehydrogenase. This is a reversible step, so it can form as beta hydroxybutyrate, or by oxidizing, it can form as keto acetoacetate molecule. So these are the ketone bodies. By this pathway, if it is not enter into Krebs cycle, these acetyl-CoA molecule enter into ketone bodies synthesis pathway in the presence of thiol-kinase, HMG-CoA synthase, and HMG-CoA lyase. So which cleave the acetoacetate and acetoacetate in the presence of dehydroxybutyrate dehydrogenase, beta hydroxy butyrate dehydrogenase converting to beta hydroxybutyrate. Okay, so this has to ketone bodies are transported. They are transported to different organs. Okay, they will convert back to acetyl-CoA. Okay, and enter into Krebs cycle. While ketone bodies thus function as alternative fuel, amino acid must be degraded to supply input gluconeogenesis when hypoglycemia occurs. Hypoglycemia means, as we see, sugar levels goes down, since acetate cannot convert into glucose. So if you see ketone bodies, excess ketone bodies can be produced in diabetes mellitus or in starvation. When excess ketone bodies are produced, we can find them ketonuria and ketonemia and ketonuria, and acidemia can result. Okay, you can see ketone bodies in urine as well as serum, and more acidic condition generate inside the system. Okay, now you can see the beta oxidation. Beta oxidation is a cyclic form degradation of fatty acid molecule into acetyl-CoA molecules. So you can see nitric acid, which is C14 acid. So it goes into 6 cycles. Okay, so 6 cycles, you can get approximately, you will get 7 acetyl-CoA molecules by the degradation, by beta oxidation of fatty acid. So if you see energy yielding by beta oxidation, take for example palmitite, which is carbon 16 fatty acid molecule. Overall fatty acid degradation of this, you can see palmitate and 8 coenzyme A and 7 NADH, 7 FADH and water molecule. You will get 8 acetyl-CoA molecule and 7 NADH molecule and 7 FADH 2 molecules. Okay, so if you calculate how much energy you will get out of this, so here 1 FADH2 molecule is equivalent to 1.5 ATP molecule. Okay, if you calculate the ATP yield from this NADH and FADH molecule I'm talking. So if you say NADH, it is 1 equivalent to 2.5 ATP molecules and 8 acetyl-CoA molecule. So once 1 acetyl-CoA enter into TCA cycle, you will get 2 ATP molecules. So 8 into 10, 80 ATP molecules. So here you can see total 108 ATP molecules will form once fatty acid molecules oxidized inside the cell. Okay, 2 ATP molecules are used to activate the fatty acid molecule. That's why you get net 165 ATP molecules out of palmitate, which is equivalent to 16 carbon fatty acid. So beta oxidation of unsaturated fatty acids, unsaturated fatty acid, which will have double bond in its structure. Unsaturated fatty acid yield bit less energy than the saturated fatty acid because they are partially oxidized. Their hydrogen molecules are already removed from the unsaturated fatty acid. Okay, less FADH2 is produced in the unsaturated fatty acid molecule. So beta oxidation of orchial fatty acids. Orchial fatty acids degrade leads to acetyl-CoA and 1 propanyl-CoA. So here you can see 1 3-carbon chain fatty acid propanyl-CoA. So propanyl-CoA is metabolized by carboxylation to methyl-malonyl-CoA. So this is methyl-malonyl-CoA and then methyl-malonyl-CoA will convert into succinyl-CoA. Succinic acid is the intermediate molecule or feeding molecule to the TCA cycle. By this way, this molecule, malonyl-CoA, propenyl-CoA, which is generated from the arch and fatty acids, also enter into utilized in the oxidation via TCA cycle. So the regulation, total regulation of beta oxidation, largely by concentration of free fatty acid available. How much free fatty acids available inside the system is one common regulation. And malonyl-CoA inhibit the carnitine transferase, which will inhibit the entry of acetyl-CoA into mitochondria. So here, the malonyl-CoA will inhibit the carnitine palmitase transferase. So that will not attach to carnitine molecule, so it will not transport it inside the mitochondria. This is one major blockage step to regulate the beta oxidation inside the cell. Okay, so now this just a comparison. You can see the carbon chain, same carbon chain of hexanoic acid, carbon structure, atom length. Okay, molecular weight, whatever you take. So hexanoic acid is C6H12O2 and glucose C6H12O6. Okay, so you can see the oxidation of this glucose and fatty acid when it is molecular weight, when carbon structure is similar. So here you can see one hexanoic acid degrades, you will get 37 ATP molecule. One glucose molecule oxidized, you will get 32 ATP molecule. Okay, so 5 ATP molecules less in carbohydrates. So that's why carbohydrates are less energy yielding macromolecule when you compare with the fat molecule, fatty macromolecules. Okay. So this is just for your reference I gave here. So Lipcott and Garnish textbooks, when you refer this one, they calculate differently how much fatty acid yields from the palmitate. Okay, so Lipcott and Garnish, they given palmitate upon oxidation of palmitate, total 131 ATP molecules are generated. Okay, 131 2 ATP molecules are utilized for the activation, so net yield is 129 ATP molecule according to Lipcott book. Okay, when you see actually I know how much it will form because of membrane leak, hydrogen ion molecules during the ATP formation, hydrogen ion molecule for ATP formation. So instead of here, they calculated 2 ATPs for FADH2 molecules, 3 ATPs for NADH molecule. Okay, because of this leakage for the ATP molecule generation in the proton gradient. Okay, so actually you will get 1.5 ATP molecule for FADH2 molecule and 2.5 ATP molecules for one NADH molecule. So taking that, calculating according to this losing the hydrogen ions during the ATP formation during the proton gradient. So totally you will get net ATP molecules are 106, not 129 ATP molecule. Okay, so upon oxidation of palmitate will give rise to net 106 ATP molecules. Okay, so that's all about today's class. If you have any doubt, please let me know. Hello. Hello.
Yes, sir.
Yes, sir.
Any
Yes,
doubt?
sir. No, sir.
Okay. Regarding your examinations, we discussed. So all faculty unanimously decided to go offline exam, not online exam. Okay, you will have online exam.
So, but for online, for the previous semester, we had at least one leave for before every exam. But this time we are having no leaves between any of the exams, theory exams.
Timetable is sent to you.
Yes, sir. Sir, all the theory exams are consecutively like 1, 2, 3, 4, 5, 6, like continuously there are theory exams scheduled.
See, thing is, as per university academic planner as well as UGC academic planner, we have to finish exams by January, somewhere before Pongal. Okay, after Pongal, we have to declare the results. Okay, based on that only timetable is framed. Okay, if you have some concern, you just send email where you want to have 1 day or break for any exam.
Okay,
Okay,
sir.
we will try to accommodate seeing the calendar and seeing that one. Okay, we are thinking to get you know some kind of permission, to extend at least 1 week. One sec. Yeah, exam is offline only. Somebody asked the exam is online, offline. Offline exam is offline only. Is it clear?
Okay, sir. Sir, is there is any change in the question pattern?
See, that is depend on teacher to teacher. Okay.
Oh,
So
okay,
you
sir.
need to contact the course teacher. What pattern they are going to give. All those things depend on the teacher to teacher. Okay, normally when we have offline exam, when we follow university pattern, we used to use standard pattern, 2 marks questions and 4 marks questions and 10 mark question like that. That will totally 60 marks. It's a 60 marks examination. Okay, so last 2 years we are following online examination. For that we are using different patterns. Somebody using short answers and multiple choice questions, different way they used to evaluate. So now we are coming back to offline exam. So just check with your course teacher what pattern is convenient, and so you can discuss with them. I cannot tell on that. Okay.
Okay, sir. Sir, and about internal two for your subject, sir.
Hmm.
So we can plan that internal two on next Tuesday, sir.
Next Tuesday.
Yeah.
Yes.
So what we are planning is if we complete the internal here, then we can able to concentrate on lab and project when we come there, and also we can have some time to study for the semester exams.
Also, as you said, we need to settle down a bit, no, sir. People are traveling from, so at least 1 week it will take.
Yeah, that's the reason we are decided. We are thinking to get the approval from the university to move the examination for 1 more week that side. You understand?
Yes, sir.
So if they give the permission, that will give, that will give buffer time to us, so that you will come and settle down and you will start your practicals and all those things. Okay, we will try my. I will try my best to get the approval from the university. No, it is not possible.
Sir, I'm asking about the internal, sir.
Yeah, internals you can take online when you are there. See, you can write if you would like to write offline. You can come and take the internals, no problem. Okay. We'll see this second internal examination whenever it is possible. We'll see. Okay. Just now I'm trying to prepare and trying to finish your syllabus. This is third unit only. We are still in third unit, isn't it?
Yes, sir.
Yes, sir.
Fourth unit, at least part of the fourth unit if I finish, so that I can take. So far we have taken first and second unit. So now third and part of fourth unit, if I combine for second internal, that will give, we'll see. Okay. Yeah, I will send. Anything else? See, one thing I would like to tell you, and please pass this to MSc students also. I'm receiving one or two mails from students that SOP undertaking forms from you.
Yes, sir.
Yeah, what I want from you is, please, collect all SOP undertaking forms and send one as a zip file. Say, for example, all MTech students, you can collect undertaking form and make a zip file and send to office address, email address. Same way, MSc Bioinformatics students also can make one folder or one file and send to office address.
Yes, sir. We will share.
Yeah, why I'm telling, today someone sending, tomorrow someone will send, yesterday somebody sent. Okay, so I will receive many mails apart from your mails also. So I may miss that one during extracting data from my emails. I may miss somebody's undertaking form. They should not suffer once they come back here.
Okay, sir. We'll do.
Yeah. So once you collect everyone's undertaking SOP undertaking forms, make a file for kind of zip file and send to contact, so that we'll print out and make everything ready, sign and all those things. Once you come, you can take your form and go and submit in the hostel office. Let's make it easy.
Sir, the undertaking form I have submitted collectively to the contact email address.
Okay, that's fine. When you submit it collectively, that is easy to us to print out.
Yes, sir. Actually, we have already submitted that.
Okay, please inform to MSc students also. They are sending me every day. I'm receiving 2 or 3 undertaking forms.
Okay, sir. We'll inform them.
All right. Okay. Anything else?
No, sir.
No, sir. Thank you, sir.
Right. You can sign off now.
Thank you, sir.
Thank you, sir.
Thank you, sir.